So basically, in fact, I stole this slide from Bob Ball. That's why I put him as a collaborator. So, and he stole it from somewhere. And this is a little old, and I think 20,000 people have used it. And vaccines, in fact, did not start with Jenner. There was a woman traveling across Europe to Turkey, and she lived there for a number of years with her husband and came back to, in the 1700s, came back to England and said, oh, I have a great inoculation thing. And she was a la mode woman, very intelligent, by the way, and she started a process that I could call equivalent to a precursor of modern immunizations. For whatever reason, we talk about Jenner, we do not talk about her. It's probably a sexist society we have, but you should read her travel books. That started vaccination in our life. There are a certain number of developments that help us improve, let's call it risk management for vaccines in the US. In the 1990s, the CDC developed the Vaccine Safety Data Link, which gave us a lot of strength in observational studies. Also, the CDC developed the Clinical Immunization Safety Assessment Centers, which help into the causality assessment. If you want, the Brighton collaboration developed as well, and now it moved more towards Europe to try to help us define what the case is, what this disease is, what this entity is, in a way that we can all use. It's a wonderful tool. And we started, with trying to generate guidances that could help everybody use the same standards. So manufacturers submitting data to a European agency or a Japanese agency or Brazilian agency don't have to present completely different things to completely different agencies with no advantage or benefit for anybody. So ICH is trying to play that role, fairly successful at this point. And we have ourselves expanded collaboration in other ways, and I will explain you how. In general, for vaccines, we have to understand the following thing. Patients are risk averse. And the problem is when you try to, let's say, eliminate measles in a whole continent, and you tell a person that the probability that his or her child will have measles is practically zero, but the probability that they will have a local adverse event or fever caused by the measles vaccine is relatively high, around 10%, the mother, any rational mother would say, why should I vaccinate my child? So, in fact, we should ask manufacturers to develop less effective vaccines, and therefore we'll have less concerns about safety, okay? So, in general, sometimes bureaucracies are a little slow in being transparent and communicate things. And for instance, we used to say vaccines are safe. Yes, vaccines are safe in relation to the benefits that they provide. So we forget the second part of the sentence, and people take us for our word and say, okay, I found that my child had fever or my child had whatever. So vaccines are not 100% safe. Each and every biologic will have problems. Our problem is what do we do with it, how do we investigate it, how serious the problem is. So, in fact, our game for vaccines, we think it is a wonderful thing. We really, all of us, depend on the public acceptance of vaccines and their perceived safety, and that is a complicated message to send. So we need a systematic approach to manage the risk or the perceived risk. And of course, as you have seen, I mean, some of you had problems traveling here because a reasonably large volcano, but not the largest in the world, exploded near Reykjavik in Iceland. I mean, why should it affect us? Well, it does. Same thing with disease. Disease affects us here, even though it originated elsewhere, in Mexico, India, it doesn't really matter. So we have a global problem, and our approach is let's take a global approach to resolving it. So we were talking about clinical trials right now, and we started, the panel discussed multiplicity. What if there is a confounder for an adverse event? Then you need twice the sample size. And what if there is another confounder? Then twice the sample size of that. Or I'm exaggerating a little bit, but relatively quick you come into big numbers. Clinical trials cannot deal with big numbers. Manufacturers are scared of big numbers. Why? Because they are expensive. Clinical trials are expensive, and we recognize that. So we try to resolve that problem. Duration of clinical trials also is limited. And also the selection of patients. There has to be some type of selection but the people who get selected to enter the population of a clinical trial are not exactly the same ones who will be vaccinated afterwards. It's not true. So what happens is we need something after the clinical trial finished to give us the assurance that the vaccine in real life situation is still safe and is still effective as we thought it was. And usually there are differences there. So we do evolve into our knowledge of the product with the use. As it was, the panel said, we have 10 years of experience here. We have this number of years of experience with this adjuvant, with this product. So we also need time, and that's unavoidable. So in an effort to help us in that, there is the Fda Amendment Act, which require more post-marketing studies. Labeling changes are a decision from the agency. It's easier for us to require it. It's easier for us to require risk evaluation and mitigation strategies when we justify its need. And we were required to establish surveillance systems for observational studies data sets for 100 million people by 2012. Can we do that? Well, maybe. So our vision, and this is just a vision which is reasonable, but it's not going to be reached in many, many years or probably ever. Ideally, all patients' biologic product exposures should be matched with their outcomes, and then we will have the perfect database to match what has happened to this vaccine and to this other in the short and in the long term. And then we will explore, I don't know, cardiac disease after the use of adjuvants 20 years later. But we will not reach that, so we'll have to be satisfied with something much more modest. So there are opportunities here in the development of health information systems, and as you will see, we are working on that, and we are extremely embedded into it. And we, of course, have to guarantee privacy in this country, in most countries, in every country, and that requires that the data is kept under the firewall of the institution responsible, and they send to us just exactly what we ask without identifiers. It's a little tricky, but it can be done. It's not so complicated once you work on it. So what our needs are: access to more patients, to better data, diversity of data sources, finding comparable control populations, nomenclatures. Brighton collaboration helps us in that, and of course, again, maintain privacy protection, which is important. Communications are important. As you see in the political game in this country and in every country, it doesn't really matter so much what the reality is, but who dominates the market, who dominates the communication. We have been trying to convince people that measles vaccine have nothing to do with autism. We have spent millions and millions of dollars in redundant investigations. Well, if you ask parents out there, probably most of them still think that measles vaccines cause autism. They dominate the communication message. The message is dominated by these type of tendencies, and we'll have to accept that that's a reality right now. We'll have to educate our population better, but also we have to be more transparent so we are trusted. When you are more transparent, people can misinterpret what you say. So it's a little complicated, but we have to play that game and do it well and do it truthfully. So I go back to this beautiful sentence. In fact, the original sentence is, a mind is a terrible thing to waste. And it was one of the sayings of the Negro movement in the United States. And an economist from Stanford said, well, a crisis is a terrible thing to waste as well. And it is true. H1N1 provided us with a challenge and an opportunity. A challenge because there were serious concerns. Oh, this mysterious vaccine for this mysterious disease. Those of you who are in the vaccine field, they will not see that mysterious vaccine there. It's just another antigen for us, basically. But you have to convince the population. But with that challenge came resources, and with the resources, our obligation to use those resources and improve the work we do. So we have used this crisis, and I hope we have not wasted it, to show that we can do much more better and more interaction with other partners and do better observational studies after a product, in this case, H1 vaccine, goes out. So we have this coordination. MVPO has a little role leading role on that, and we are working under the umbrella of a national vaccine advisory committee, which keeps us honest, if you want. So basically, in pharmacovigilance, there are no mysteries. What we have is what we traditionally used until recently, which was passive surveillance system, vaccine adverse events reporting systems, and systems like that. Those are very, very imperfect tools that are only an alarm signal. And I will use something that I have used in the past, so if somebody saw any of my presentations, assume that I'm not repeating myself. When you have a house alarm, the house alarm is there to protect you from burglary. But the problem is 99.99% of those alarms, when they sound, they will not reveal a burglary. A cat, a neighbor, your child, etc. But the police have to go anyway. That is what a passive surveillance is. It's extremely sensitive, completely non-specific. So on and by itself, it is really a waste of resources, an enormous waste of resources. So we have to go into a strengthening and confirmation of those signals by more elaborate tools, doing observational studies, knowing your population, increased communication, collaboration between institutions, so we compare the results we find here with those we find here and there. Collaborate internationally, of course, sharing safety data. So we have used that. We have used the resources given to us to work more closely with the Department of Defense, with the Medicare services, with the Indian Health Services. This is misspelled. The computers nowadays do not accept IHS. Whenever you write IHS, the computer writes for you HIS, and I have to justify the mistake all the time. I love the IHS people, but I'm sorry for them. So Sentinel Activities, as we said, from Fda, and a stronger international collaboration, which is not so complicated as it will seem. I will give you an example. So briefly, vaccine adverse events reporting system, it's just an alarm signal. The fact that there is a temporal association between my child vaccinated and he having an accident 5 minutes later when he left the clinic could be coincidental, or could be just that this child, who is 15-year-old, just took his father's car and drove out of the clinic and had syncope. So it doesn't prove there is no association, but it doesn't prove there is association here. It's just a signal. We have to go much, much deeper. The PRISM, which is part of the surveillance system, the Sentinel surveillance system, our collaboration with electronic systems, strong electronic systems which have good denominators. The Department of Defense, 1.4 million active duty personnel. We have access to all that data without identifier for us. We have the same with the Veterans Administration, elderly military veterans. Same thing. We are working with less than 1 million, but we will work with more in the future. CMS, an electronic database, but no access to medical records for a number of reasons, but 38 million people. So we can do timely surveillance and detect adverse events in time. The problem is, the more you demand from a system, the more mistakes you will get into, and the more you will have to verify. The more questions you ask to a system, the more answers you will get, and some of those answers, you will not like them. Just by chance alone, if you try to look for 20 events, one of them is sure to be positive, regardless of real association. So it is always a problem. But we try to also work with international partners. How do? We try to move our partners from passive surveillance into more active surveillance, into something that can be done analytically. So I will give you an example, a GBS study on the association between Guillain-Barré syndrome and H1N1. Using what? Using hospitals, using only numerator data. As Archimedes said, if you give me a lever, I can raise the world. And it is true. If you give me a few hospitals, I can give you a study. And it will be an observational study, beautifully done, insofar as we have a risk window to compare with a non-risk window. We just go classify the case, define what the case is, whether it is, in this case, GBS or not, in which week it occurred, compare the incidence with the incidence in a non-risk window, and that is, voila, the analysis. So give me hospitals, I will give you analysis. It's simple. We can universalize any question. I was talking to our Spanish collaborators today. I said, we are working with the GBS project. And one of them says, oh, we are interested in HPV. Okay, I don't care what you put in my table, just give me the name of the disease, the risk window, and the comparison windows we will elaborate together, and we will do the analysis. It's not so complicated to collaborate, but you need to be sure not to bias the study because of the way you search for the data. The rest is really simple. So basically, vaccines have real risk. We have to take them seriously, particularly serious adverse events. Effective vaccines do automatically increase the demand for safety, and there is no way out of it. The better vaccines produce herd immunity, and we will have to live with it. An evaluation system needs pre-marketing evaluation, detection of signals, and verification. There is no way out of it. And agencies have to collaborate to understand the biology and to define the risk and the benefits of each product, and we have to communicate with the public. So it's really not complicated, but here we are after 200 years of the beginning of vaccines and still questioning them. They are a wonderful thing. We have to keep them safe. Thank you.
Lance Gordon, Immunobiologics. Could you comment on interagency cooperation? Is FDA able to draw on NIH laboratory resources to look at mechanisms that might be associated with adverse events and, say CDC monitoring programs? Are we capturing biologic materials that can feed into all this? How do the different agencies fit together?
Right. That's a simple and complicated question for a number of reasons. Privacy is always a concern for any regulatory agency and any research institution. So when we are talking about us requesting a physician to request his or her patient to produce biologics material because we want it for our research, the ethics requirements for that are extremely high. Now, CISA has been developed by CDC with that exact purpose. And of course, scientists from the CISA group and from the CDC laboratories and from the FDA laboratories work together with NIH laboratories when and if needed. But this is a general optimistic we work together answer. It is extremely, extremely complicated to work with biologic samples. I don't know if Dr. Modlin has any comment. You have worked on that, but it's complicated. I love it to be simple.
Just a simple question. Post-marketing surveillance basically is phase 4 clinical. How difficult would it be to do phase 4 clinical for vaccines?
Well, we do phase 4 clinical studies for vaccines. In fact, we can require manufacturers to do what we call post-marketing commitments and post-marketing requirements. Now, the problem with this, everything is a very complicated dance. Basically, if we are developing our own systems, our own observational systems, as we say, with collaborating with other partners, federal partners, et cetera, private contractors. We cannot ask the manufacturers to do a phase 4 study when we do have the resources to do it ourselves. So if we have the resources, we do it ourselves. Now, when we cannot do it ourselves, we ask the manufacturers to do a study. Now, you cannot just say, it's Friday, 7 p.m., I want to ask you a 50,000 or 100,000 or a 500,000 people study. You have to justify it, and that's when you go into the commitments of the manufacturer and what you can effectively require. The commitments are voluntary. The requirements, the post-marketing requirements, we can enforce them, but we have to justify them. Same for risk management strategies when you have smallpox vaccines and things like that. So yes, we can do it, but everything goes into the negotiations dance, and you have to be careful with that because you also want vaccines in the market because it benefits the public. And the more competition we have, the better off we all are in regard to vaccines that are accessible for us. So it's complicated, but yes, we have the tools to do that, and those tools are stronger for us now. Okay, thank you.